Hello, and a very warm welcome to our Cell and Gene Therapy Insights webinar on increasing productivity of IVT reaction using at-line monitoring by HPLC chromatography. I'm Elisa Minotti, founder of BioInsights, and joining me today is Rok Sekirnik from BIA Separation, part of Sartorius, who will be discussing the application of an HPLC-based technology using a multimodal ligand based on anion exchange ligand to monitor the IVT reaction. The method allows for simultaneous quantification of NTPs, capping reagents, plasmid, and mRNA within 3.5 minutes. So following Rok's presentation today, we'll have a Q&A session. And as always, do feel free to ask a question at any time throughout the event, just using the Ask a Question box, and we'll try to get during the session. But before we get started today, I would just like to draw your attention to the resources tab. You can also find some links to documents to get further information on the topics that will be discussed today. And now I'd like to introduce to you our speaker, Dr. Rok Sekirnik, who is the Head of Process Development in mRNA and pDNA at BIA Separations, part of Sartorius. He graduated in chemistry at the University of Oxford, where he also completed his PhD. And he has worked in the areas of monoclonal antibodies, vaccine and gene therapy vector development in various roles. His team at BIA Separations is working on developing high-yielding, cost-effective purification approaches for pDNA and mRNA production. And so with that, I will hand over to Rok for today's presentation.
Thank you. Thanks very much for this nice introduction, and welcome everybody to this webinar. I hope my pointer is working, and so I can take you through the presentation, which will span the topic as announced. So at the core of it will be the question of how to increase the productivity of IVT reaction using HPLC tools, which I'll describe in detail. But I would also like to understand this question broadly, and so I would like to consider in this webinar also factors that feed into the IVT, such as plasmid production, plasmid quality, as these are at the moment, I think, some of the very important and hot questions in the field of mRNA production. What is the right purity of plasmid and how to make it, how to purify it, and then I would also like to extend the productivity of IVT beyond the IVT reaction itself to the actual purification, so that we don't lose too much material, valuable material, once we've made it. So this is the scope of the webinar today, and I like to start every introduction by considering some of the essential properties of mRNA. We can say RNA, as the M is being joined by other acronyms of recent, and we look at it from the perspective of physical chemical properties rather than biology. So our task today is to produce and purify mRNA drug substance rather than consider its in vivo properties. So we like to consider things like size, charge, and any affinities that RNA would have. And so the very first consideration, which is oftentimes forgotten, is just how large even a small mRNA molecule is compared to standard biologics. If IgGs were considered relatively large back in the day at 150 kilodaltons, the mRNA vaccine, which a lot of the world has been vaccinated with, encoding for the spike protein, well, that is around 1.3 megadaltons large, so nearly 10 times as large as an IgG. This should put it by all means into a different category of biomolecules, bringing with it both advantages and opportunities for purification, as well as, of course, significant processing challenges, which I'll address later on. But one of the great features of RNA, of course, is also the ability to encode sequences into it. So one of the most desirable sequences that mRNA can have is the poly-A tail, from the perspective of purification, that is, as well as in vivo stability, and this can be very well utilized for affinity purification. If I link it back to IgGs, it is very homologous to protein A purification of IgGs, for example. If we think about the charge of RNA, well, we learned early on in high school that mRNA is composed of the phosphate backbone, and this phosphate backbone remains negatively charged down to the lowest of pHs. So the mRNA molecule is highly negatively charged, so it will bind to anion exchanges. But there can also be hydrophobic properties to mRNA if exposed to the right conditions. It can actually exhibit significant hydrophobicity, and this we can utilize for binding to hydrophobic interaction media or reverse phase media. So these are some of the key features that I'd like to take with us as we travel along the journey of producing and purifying mRNA. And then there are, of course, the more strictly biochemical, critical quality attributes, if you wish, which relates to the mRNA sequence and capping properties. So we know that the 5' cap needs to be carefully engineered to prevent in vivo degradation, and here we use capping reagents or enzymatic capping to install the correct capping form as the very first nucleotides in the sequence. And I mentioned the poly A tail, which is also needed for in vivo stability. And in between is the coding sequence, which of course is the magic of every therapeutic RNA sequence. And as we produce mRNA, we actually also can produce a lot of contaminants or bring with the raw materials a whole host of contaminants, which we then in the process need to be removing. So in the course of this presentation, we'll also meet some of these impurities which stem either from the raw materials used for producing mRNA, and that can come from the contaminants related to the plasmid, which is typically produced in bacteria, and so a lot of bacterial contaminants need to be considered when thinking about the purity of mRNA, as well as the impurities that are produced as part of producing the mRNA in the IVT reaction, and these are double-stranded RNA, can be RNA fragments, abortive transcripts, and so forth. So this is the list of all of the challenges, let's say, that we need to consider as we develop our mRNA process, and a lot of the answers or the technical solutions which I will provide in this presentation will be based on chromatography, because chromatography has been historically one of the most selective and scalable tools for purification of biologics. And as mentioned, as the very first concept of mRNA, its size, the chromatography itself needs to be carefully designed to match this large size of mRNA, meaning that chromatography, as has been traditionally employed for purification either of small molecules or proteins, which was based on porous particle based separation, no longer necessarily fits the paradigm of mRNA for multiple reasons. One is the small size of the pores into which a molecule as large as mRNA cannot easily enter. The second is that linked to the large size is the low diffusion, the slow diffusion of this exceedingly large molecule. And even if the pores on the porous particles were large enough for the mRNA to enter, the diffusion into those pores would be very slow. And on top of that, the flow properties within or amongst porous particles are such that a lot of turbulent forces are created, and this could be detrimental to the intactness of mRNA. So shear stress could be problematic for the intactness of mRNA. And so we need to use a different chromatography concept, one that is better suited to molecule of the size of mRNA, and this is where chromatography resins based on wide interconnected flow channels that are modified with suitable ligands come into play. And when I say this, I really refer to monolith chromatography, monoliths being typically polymethacrylates, polymer-based chromatographic devices with interconnected flow channels of the size from between 1.3 micron to 2 micron, ranging all the way up to 6 micron for very large plasmid DNA or mRNA, which can easily accommodate a molecule of the size of mRNA, which can bind mRNA based on convective mass transport rather than diffusive mass transport, meaning that we can easily deliver the molecule to the binding sites with flow and then dissociate it immediately and not have this movement based on diffusion. It is a chromatography resin where turbulent forces are minimized, the shear is minimized, and so there's very little concern that any shear forces would damage the mRNA. And also the walls of this monolith, as I show it here on the far left of the slide, can be modified using pretty much any ligand we can think of. And indeed, we have modified monoliths with a whole host of different ligands and are continuing to do so to get to better and better and better ligands, better suited to new modalities that are emerging. Today we'll be primarily discussing affinity ligands, so oligo-DT-based ligands, as well as some multimodal ligands that combine elements of anion exchange and hydrobonding, which are particularly suited for purification and analytics of mRNA. And we'll use those ligands in a range of steps, and this is the first slide where I would like to very briefly address the structure of a typical mRNA drug substance production process. I don't address the fermentation, but I address all the steps that are downstream from fermentation, namely cell harvest and alkaline lysis, which are designed to open up bacterial, typically E. coli cells, to release the plasmid, which encodes for mRNA. And we need to then purify the plasmid from host cell contaminants, such as host cell DNA, so genomic DNA from E. coli, host cell proteins from E. coli, endotoxins from E. coli, and ideally also isoforms of the plasmid itself. And all of this we do in the top line of this process, at which point, after linearization, we arrive at the linearized plasmid DNA, which we then take into the IVT reaction, which I will describe in more detail later on. And the product of IVT reaction will purify either with affinity or with this multimodal ligand to capture out the mRNA. And if necessary, we can then polish it using a host of tools before we do buffer exchange infiltration to arrive at our mRNA drug substance. As I said, the first component of every IVT reaction is the plasmid. So I would like to very briefly touch upon some of the critical aspects of the plasmid and in part respond to some questions that we often receive about what is the purity level that's required of the plasmid and how to meet the purity levels that are either stipulated internally by developers and producers of plasmid or by external bodies. And I think the fair answer is that there is currently not very clear guidance on the required purity for plasmid to be used as a raw material for production of mRNA. The regulatory specifications for the use of plasmid as a therapeutic agent itself are clearer, but those shouldn't necessarily apply to the plasmid as a raw material, because there's, of course, multiple downstream processing steps still following from the plasmid. Nonetheless, one of the critical questions that we get is, what is the necessary level of purity of supercoiled plasmid versus open coil? And I want to spend a few minutes on this question in a minute. But before I go there, I want to just mention what are the key purification steps to separate out plasmid from other E. coli impurities. And this is namely weak anion exchanger, diethylamine ligand, that works very well in the processes, is extremely well understood and templated, let's say, for a whole host of plasmid sizes. And you can purify plasmids up to 10, 15 kilobase pairs using process steps that are prescribed. And in this process step, you will isolate the plasmid and remove the majority of host cell proteins, genomic DNA, and host cell RNA. And then this will be followed either directly by linearization, or if need be, and if internal specifications so require, the plasmid can be directly purified by hydrophobic interaction chromatography C4 or butyl column to enrich for supercoiled isoform and remove endotoxins before going into linearization. So I don't stay at the abstract. I want to show real examples of purification of plasmid that will then, in fact, also be used for IVT. So we'll be making mRNA from this plasmid. In this plasmid capture step, we apply the DEAE column with a gradient of salt, which is very well understood, to remove the RNA with the so-called RNA wash, and then we perform the linear gradient to remove any residual RNA that was not removed by the half-molar sodium chloride wash, and followed by elution of the plasmid DNA. We monitor this with our platform that we call PatFix HPLC platform, utilizing the CiMac pDNA column to follow the purification through. In fact, all the way from lysis, filtration, DEAE, load, wash, and elution, till we removed, as you can see here on the left hand side, at approximately 4 minutes retention time, we removed the RNA in the RNA wash step, and upon elution, we obtained peaks that correspond to supercoiled plasmid, open coil plasmid here on the left, and a little bit of multimers that stem from the specific sequence of this plasmid. So typically, we would recommend that this level of purity is sufficient to proceed into linearization, and we can have a certain percentage of open coil isoform present. But more and more, the community is asking for plasmid of over 95% supercoiled purity, and we get a lot of questions how to purify out the open coil. So I want to show an example. This is not the same plasmid, but the concept applies just as well on using then this hydrophobic interaction chromatography as the subsequent step to polish out supercoiled plasmid if the requirement is 95% purity or more. In fact, in this particular experiment, we managed to get pretty much 100% pure supercoiled plasmid, as you can see here on the right-hand side chromatogram. But the mode of utilization of hydrophobic interaction chromatography is slightly different from the bind dilutes, which we would typically use. With bind dilutes, you can achieve 85% purity of supercoiled, but with this way of using it, you can increase the purity of supercoiled above 95%, and it's It's called sample displacement chromatography, which in its core means loading the C4 column at a condition at which open coil does not bind and supercoil binds, and load the column until the point of supercoil starting to break through. So what we've done is we found conductivity or concentration of ammonium sulfate at which open coil didn't bind to the C4 column, supercoil did, and we went up to the point of saturation of the column with supercoiled and achieved essentially the purification of open coil out of the sample. So this is enriched open coil structure here in the flow-through fraction, and elution was pure supercoiled plasmid. So if for reasons of risk assessment, let's say a company or a laboratory wants to stipulate for itself a 95% pure supercoiled plasmid for downstream use in IVT, this is perfectly possible. Although I should like to add my personal opinion that the percentage of open coil, to my knowledge, has not been shown to be detrimental in any way to the quality of mRNA produced. And so I welcome actually an open discussion or a demonstration that percentage of open coil might affect the quality of mRNA produced. But so if experimental data comes out to suggest that the purity of the supercoil needs to be as high as 95%, there are tools available to achieve that. The next step, and we're traveling now very quickly towards the IVT, is linearization. And I want to add that same tools as we were seeing earlier on monitoring the purification of plasmid to obtain our supercoiled plasmid can be used also to monitor linearization. In fact, this is one of the critical steps from the point of view of production process to understand the linearization kinetics. What's the minimal concentration of enzyme that's required to linearize a certain mass of plasmid? And so while agarose gels or capillary electrophoresis are useful, they're not easy QC tools to use or interpret. HPLC is a workhorse for every QC lab, and so we highly recommend the implementation of the CiMac pDNA method, which very nicely resolves open coil isoform from linear, from supercoil, from multimers, and so we can plot the peak area as a function of time to monitor the progression of linearization of the plasmid, and then start asking process-related questions such as what's the impact of temperature, what's the impact of pH, what's the impact of enzyme concentration, what's the impact, for example, of additives such as BSA. Does BSA really have a significantly positive effect on linearization? And this is in fact the question that we asked in this particular experiment, and the answer was there was a mildly positive impact of BSA, but the linearization was completed in just 2 or 3 additional minutes in absence of BSA, so it was completely safe to drop BSA in this process, and this was information that was quantitative and could be easily interpreted. So finally, we have produced plasmid that we can now take into our IVT reaction and start optimizing the IVT reaction. So let us consider what's the job ahead. IVT reaction is an enzymatic reaction of converting double-stranded DNA that we've just produced and purified into mRNA or other RNAs, and it typically utilizes T7 polymerase and NTPs, nucleotides, as the building blocks of RNA. As mentioned earlier, RNA for in vivo application requires 5' capping, and the cap can either be added co-transcriptionally, meaning in the IVT reaction, or post-transcriptionally, where we use enzymes to install the 5' cap. And then there are additives to the reaction which can enhance the yield. It's a multi-component reaction. So how do we go about optimizing this and not get lost in the optimization? Well, there are nice publications describing DOE approaches, and they quote something like 8,000 possible combinations of all these factors. So it's too much to handle in a single factor at a time approach. So DOE is probably going to be the workhorse from the theoretical perspective. What about from the measurement perspective? What's the best measuring tool that we can apply here? And it is very much our view that what's important as a readout in the reaction is the concentration of mRNA, of course, as the measure of productivity, but also the concentration of nucleotides. On top of this, there's value added in measuring the concentration of capping reagent, being one of the most expensive reagents in this already expensive reaction, and potentially also double-stranded RNA content is one of the major or most critical impurities. And so I want to jump into this by presenting 3 different analytical tools which we can utilize to monitor these outputs. The first one that we'll address will be the very simple affinity-based analytical monitoring of production of polyadenylated mRNA. Second will be a method that can actually track mRNA as well as NTPs and capping reagent. And I'll then also very briefly touch upon a method that can also track the concentration of double-stranded RNA. I should like to say that we can do this in different formats. A very new format that we've developed is a 96-well plate, where you can use oligo-DT ligand in a 96-well format to increase the throughput of analytics, or for an at-line UV measurement of the elution fractions, we highly recommend CiMac Analytical Monoliths coupled to the PatFix HPLC platform. Why we like using monoliths and why they have such nice properties for mRNA. I'm borrowing slides from many years ago when their chromatographic properties were profiled on IgGs, where we've shown that monolith, because of its convective mass transfer properties, exhibits flow rate independent chromatographic properties, meaning resolution as well as binding capacity are flow rate independent. And from analytical perspective, that means we can increase the flow rate to the pressure limit of the system and not sacrifice resolution. And that becomes very important when we try to have very rapid feedback on our IVT reaction. So the first example of this is a very simple oligo-DT affinity analytical method. Perhaps some of you are familiar with the protein A analytical tool that's used for quantification of IgGs in the monoclonal antibody production processes. This is its brother or sister in the mRNA world. So if the mRNA in the IVT is polyadenylated, we can very quickly quantify the content of this mRNA in the IVT reaction mixture by binding it to the oligo-DT analytical column. Anything that is not polyadenylated will flow through, and polyadenylated RNA will bind, and we can elute it selectively and measure the area under the peak, compare it to a standard curve, and read off the concentration of RNA at this particular point in time in this reaction. We demonstrate that this is a highly reproducible, highly accurate concentration, but it is monitoring only one parameter, namely concentration of mRNA and nothing else. So what about if we want to track RNA that's not polyadenylated? And what if we also want to collect data on nucleotide concentration at any one point? Well, that's when we need to switch the ligand on the monolith backbone to multimodal ligand combining elements of anion exchange and hydrogen bonding that we called Prima S. And Prima S has a very high selectivity for mRNA over plasmid DNA and over NTPs, and we can nicely separate out GTP from ATP from a peak that combines both UTP and CTP, which is then in turn separated from various capping reagents. I show here examples of CleanCap AG and ARCA. CleanCap GG can also be separated at a discrete retention time. So you can profile all of the key ingredients, components of the reaction that will make up the RNA structure, the RNA sequence. This is UV detection, so as long as the UV detector is well calibrated, the responses will be linear over a wide range of concentration. So if we have a tool that can and I should also stress that the time axis here, in case this is showing small on your screen, is 3 minutes. So within 3 minutes, we've read out the concentration of RNA and the concentration of nucleotides. The whole method is then additional few minutes long to re-equilibrate the column and get it ready for the new cycle. But I think the whole method takes something like 8 minutes. And in the next 8 minutes, you can load the next time point and next time point and next time point again. So you can start playing with the IVT reaction and start asking questions. And the most basic question is, why did my IVT reaction stop? And the answer, most of the time that we see, is simply because we ran out of one of the nucleotides. Running out of one nucleotide is, of course, a sufficient reason for the reaction to stop. Without this tool, it's quite difficult to know which one it is, and also what is the rate of depletion of each nucleotide. So by monitoring or by integrating the peak corresponding to mRNA, shown on the right-hand side graph under the label mRNA, you see a nicely increasing peak corresponding to mRNA, and next to it you see a nicely decreasing peaks corresponding to individual nucleotides. We can then turn this into numbers, and so we can plot out concentrations of individual nucleotides as a function of time, and we see that in this particular case, ATP was consumed first. Its rate of depletion was fastest, followed by GTP, followed by the peak that combines the concentration of both CTP and UTP. So we can then design the next iteration of this reaction, either increase the concentration of ATP or increase concentration of all nucleotides, adjust this based on the sequence of the RNA, and so forth. So a lot of possibilities to optimize the IVT just from the concentration of nucleotide perspective. But as we said, there are as many as 13 different components of the IVT. One of them, and this is already covered quite nicely in the literature, is magnesium. But the literature doesn't quite agree on what's the optimal concentration of magnesium. And a logical conclusion is, well, there may be a different concentration of magnesium needed based on other parameters as selected. So we've taken one example, one mRNA sequence under particular IVT conditions, and we've titrated magnesium from 6 millimolar to 50 millimolar. And we find two things. One is that the optimal seems to have been for this particular reaction between 12 and 25 millimolar, but also that the kinetics of production of mRNA differed significantly between those reaction conditions. So if we took an endpoint measurement, which is oftentimes the case in standard mRNA production workflows, and if this endpoint was at 3 hours, we would lose a lot of the kinetic information that's hiding for us here between 0 and 1 hour time points. So sometimes we actually find conditions which give optimal yield significantly faster than the expected time point. So if a protocol suggests leave the reaction for 2 hours, we often find that the reaction actually plateaued after half an hour, and that leaving the reaction for up to 2 hours is actually starts to be detrimental to the quality of mRNA. So we've now optimized just one factor at a time, but we've gained a lot of insight into the impact of this particular factor, namely magnesium, on the kinetics of mRNA production. So we can make decisions about which magnesium concentration to take forward and what is the selected time point from this point onward at which we'll be monitoring our mRNA productivity. And the second paradigm is, of course, feeding the reaction. Now, what are we going to feed and when are we going to feed it to increase productivity? That's quite challenging in absence of monitoring tools, but with a monitoring tool that gives a response within 3 minutes of injection, that becomes more feasible. And so we can actually run the reaction turn the reaction from standard batch to a fed batch reaction by carefully monitoring the rate of consumption of NTPs. And in this particular case, we've set a threshold of 20% remaining lowest nucleotide before we applied a new feed. And we fed the reaction at multiple time points. This was done manually. The next logical step for this is to automate this, for example, in an Ember platform, where we then turn the rate of consumption of nucleotides into a continuous feed addition. But we have to take into account other properties of the reaction, namely that the rate of consumption of nucleotides slows down with time, presumably due to also pH effects and saturation of the solution. What's the use of this tool? So not only did we increase the productivity from, let's say, standard 5 milligrams per milliliter up to over 10 milligrams per milliliter, and actually because of the feeding that we've increased also the milliliters or liters, if you want, and so that the productivity in absolute terms has increased really significantly. We can turn this into a continuous operation. We can also use the tool to monitor scale-ups. We can use the same tool for tech transfers to really understand, is the IVT performed in the same way at different scales, at different sites? Is the kinetics of production the same? An important question here is, so if we can take a readout every 3 or every 10 minutes, let's say, to be very conservative, what about monitoring the reaction, let's say, every 1 minute or so? Well, we have tested freeze-thaw stability of these samples. So if you were to take samples at every minute, freeze them, you can then analyze them offline and let's say fit the data back into the kinetic plots. So that's also possible. But if you want to respond to an ongoing reaction, then the recommended sampling time is approximately 10 minutes. And then you can respond to the reaction progression in real time. One of the key quality attributes is also capping efficiency, and there are very sophisticated mass spec-based techniques to determine capping efficiency of IVT reactions. We are developing, this is ongoing development, methods that are based just on enzymatic digestion coupled with LC UV-based quantification of mRNA, based on very simple concept that's been published before us, the fact that digesting the mRNA with polyphosphatase followed by a terminator exonuclease digests any uncapped mRNA. And so then by definition, the remaining mRNA is capped. And so we can apply this concept then to measure the concentration of our capped mRNA in the reaction. So how do we do that? We use the same analytical method as we just used for tracking the productivity of IVT. The IVT product we digest with polyphosphatase and exonuclease and inject it back on the Prima column with the same gradient, same analytical settings. The result looks a little bit like this. So we get some remaining mRNA. This is mRNA that was not digested by the exonuclease. Any uncapped mRNA ends up as a digest product, and the enzymes used for this reaction occur in the flow through of this reaction. So if mRNA was 100% capped, then we get no digest product, we just get mRNA. If mRNA was not capped, then this peak disappears and the digest products become obvious. So we have analyzed here, for example, 3 mRNA sequences capped with clean capped AG and determined really high percent of capping efficiency on those mRNA materials. When we compared it to mRNA that was not produced with a capping reagent, the RNA was completely digested. And so this is an indirect validation of the method, which we then extended and coupled with the measurement of IVT productivity. So to add the remaining dimension being the capping efficiency. So this is perhaps a busy slide. What it tries to say is that if we combine the elements discussed up to now, so performing IVT reaction either in batch or in fat batch, and considering also capping efficiency, we can now combine the measurement of productivity as well as capping efficiency. And so I give here a nice example of comparing mRNA produced with ARCA capping in batch, meaning we've just produced the recipe and didn't feed any additional nucleotides and respected the 1:4 GTP to ARCA ratio, which is known in literature to lead to highest capping efficiency. Or we've continuously fed the reaction with nucleotides while keeping the ARCA to GTP ratio constant. And that way, what we've demonstrated is that while in both of these cases we achieved nearly 80% capping with ARCA reagent, the productivity of the reaction was significantly higher in a fed batch mode. So we produced nearly 12 milligrams per milliliter, whereas in a batch simple recipe mode, you would produce something like 2 milligrams per milliliter. So you can really play with your system based on the setup that you've got, based on the capping reagents that you've got, and based on the requirements of the particular capping reagent. So ARCA requires this 4:1 ratio, clean cap doesn't, and so forth. So you can really design your IVT procedure based on solid analytics that gives you the concentration of mRNA, the concentration of NTPs at each time point. You can then use the terminator exonuclease to digest off any uncapped product to also read off the percent of capping of this RNA. Right. I feel like this was now a very rapid insight into optimization of IVT. So allow me in the remaining few minutes just to fly through what we can then do with the mRNA that is so thus produced. One of the most burning questions in the field is, what about double-stranded RNA that we also produce? And I alluded earlier to one of the analytical and preparative tools. This is reverse phase chromatography. The column is called SDVB, or styrene divinyl benzene, and I get a I should get a point for every time I pronounce this correctly, and we can nicely validate the elution profile from SDVB chromatogram with dot plot showing that the main peak typically corresponds to single-stranded RNA, and peaks eluting later correspond to materials that also give a signal on J2 dot plot, meaning they correspond to double-stranded RNA. And this same chromatography mode, namely reverse phase chromatography, would also be used if you wanted to analyze fragment size of mRNA produced in an IVT. So this has a very high resolution between different sizes of mRNA produced. All of this is packaged inside of a software that is offered as something called PatFix HPLC platform. So the Prima S method, the oligo-DT method, the SDVB method, that can all come pre-programmed in this HPLC platform. If you're interested in this, please reach out. Or if you have a HPLC system and just want to utilize the column, those are also self-standing columns as well. So with that, we've completed some part of our mRNA production process. We've now successfully produced mRNA in the IVT reaction. I would just like to remind us, and then move to questions, that there are then multiple ways of purifying the mRNA. Traditionally, this was precipitation. Sometimes you also see tangential flow filtration, and chromatography being one of the most scalable and selective purification tools that addresses, as already alluded to, multiple of these challenges presented by mRNA, namely size, shear sensitivity, stability, and ease of operation, really. And we provide a number of solutions, of which I already addressed: oligo-DT and Prima S and SDVB, and C4 only in the context of plasmid purification. So I would like to fast forward, in fact, to your questions. So allow me just to finish off with 2 or 3 thoughts based on the purification of things that we've been learning over the course of the past year or so, namely that affinity purification by oligo-DT is not only scalable, and we've been working hard on increasing the capacity of oligo-DT for purification of polyadenylated mRNA. Just this week, an application note has gone out that shows how to achieve capacity of 6 milligrams per milliliter using particular binding conditions. And this is not only a nicely scalable approach, it's also one of the few approaches that leads to a long-term stable mRNA drug substance. Compared to traditional precipitation techniques, we find that oligo-DT tends to preserve long-term stability even at elevated temperatures, such as 37 degrees, when mRNAs purified by other means tend to degrade. We've shown this not only on a let's say standard EGFP mRNA, we've also shown it on a much larger molecule called Cas9, which is a 4,000 nucleotide molecule, which behaved much the same. If purified by oligo-DT, it was stable, in this case, up to 14 days at 37 degrees, whereas precipitated material degraded. And for those of you who produce non-polyadenylated mRNA, or for different reasons don't want to go down the affinity route, Prima S, so the workhorse of our analytical workflow, is also very useful preparative tool. It will selectively purify mRNA, irrespective of whether this is polyadenylated or not, but it operates in a slightly different purification mode, namely typically with a pH gradient. And so there we sometimes get questions, what about the impact of pH on stability of the material? And we very much see experimental evidence that mRNA produced also by pH gradients on multimodal exchanges, if neutralized immediately, are stable at room temperature even at 37 degrees for several weeks. And so chromatography, we believe, is really the solution to addressing issues of sensitivity, scalability, purity of mRNA drug substance. And so I finish off with this slide that shows a calculation of amounts of materials that we can produce in a single run, which should address the biggest needs of mRNA production. With that, I conclude. I thank you for your attention. The take-home messages I will leave on the slide. Some of what I discussed today is also covered in a handbook called Purification of Nucleic Acids, which our CSO Pete Gagnon published a year or so ago. It's a very useful handbook. If anybody's interested in reading the contents, please reach out to us and we'll be glad to share it with you. And with that, thank you for your kind attention, and I would very much like to engage in a discussion.
Thank you very much indeed, Rok, for a great presentation there. And yes, indeed, if you'd like to submit your question to Rok, please do so using the Ask a Question box, and we'll try to get to it in the next 10 minutes. So just moving on to our questions then. First of all, does the method work for non-polyadenylated constructs?
So I assume that the question relates to the Analytical Prima S method for monitoring the IVT productivity, and the answer is very much a solid yes. It works for polyadenylated, non-polyadenylated mRNAs. It works for other RNAs as well.
Fantastic, thank you very much. And the next question that's come in, so what is the size range of RNAs that can be applied?
We see a whole range of RNAs being used using this analytical approach, 1,000, 4,000, 6,000, 8,000. I hear reports of larger even than that being used around the world. So theoretically, there is no limit to the size of RNA that can be used with this approach. Yep.
Excellent, thank you. And next question, can you monitor various capping reagents?
Yeah, that's a good question. In one of the slides, I've shown a chromatogram that distinguished between ARCA capping or CleanCap AG. I didn't show the example for CleanCap GG, which would also be baseline separated for those two as well as from the nucleotides. So for all of the major, let's say, clinically relevant capping reagents currently used, you can use this method to quantify also their content in IVT reaction, yes.
Fantastic, thank you. And moving on to our next question, can modified NTPs also be monitored by the Prima S?
So we have demonstrated the use of pseudo UTP, which in fact, interestingly, has the same chromatographic properties as, let's say, wild type UTP. And this is probably one of the most relevant, clinically relevant nucleotides. But any modification of nucleotides could be analyzed. We have not yet had the chance to profile all of the different variants, but they would all bind and presumably separate in the order of elutions that's close to what we saw in the slides, namely variants of GTP would elute under the GTP peak, ATP under ATP peak, and so forth.
Excellent, thank you. And our next question, can a quaternary HPLC be used for the Prima S analysis?
Yes, in fact, that's a stipulation. Quaternary HPLC should be used for this analysis, mostly from the perspective of the need for regeneration of the column between the runs.
Great, thank you. And next question, can IVT for production of mRNAs of different sizes be monitored by the Prima S?
Yes, I think that's closely related to one of the first questions. The answer is yes. So we can we've currently, again, spanned the range of 1 to at least 8,000 nucleotides, and 10,000 should also fit. And if you've got a larger one, please reach out, and we'd love to hear from you.
Fantastic. I think we have time for a couple more questions from the audience. Do you recommend monitoring the IVT reaction of a production run that has gone through process development to optimize the IVT reaction? Is there enough batch-to-batch variability that requires monitoring? And if so, how frequent should monitoring be done in a production batch?
That's a great question. I would certainly recommend monitoring the reaction batch to batch to ensure reproducibility. So depending on what are the currently monitored quality attributes, content is typically one of them, but the way the content is measured is sometimes questionable. Using this technology to monitor the kinetics of production really ensures reproducibility batch to batch. So if there is a reagent, perhaps that has an issue, this will be picked up by the kinetics. If T7 polymerase, for example, has undergone any kind of quality incident, the reaction will be slower. This will be picked up early on and marked as a deviation, so you can actually respond to a quality event earlier. And of course, this is an IPC, so it's an in-process control method, and I would certainly recommend monitoring production runs, at least for info only, and perhaps in the future also as part of batch records.
Yeah, that makes a lot of sense. Thank you. And just one last question to recap: what size Prima S columns are currently available?
So we've certainly can supply columns up to 8 liters available in GMP. 40-liter columns are also in our production facilities. If this is the production volume that you would need, please reach out, and our commercial team will be glad to hear from you.
Fantastic. Well, that takes us very neatly up to the hour, and thank you so much, Rok, for a great presentation today, and also for an enjoyable Q&A session. Thank you to our audience, as always, for joining us today and for your questions. The on-demand of the webinar will be available tomorrow, so look out for an email from me in your inbox, and obviously do feel free to distribute that amongst your colleagues. And just to say thank you again, Rok, for a really enjoyable presentation today. And yeah, I hope you can join us next time for our next webinar. But thank you very much indeed for joining us today.
Thank you. Bye-bye.
Thank you. Bye-bye.